Hello, I'm Nicholas Fernandez. I'm a computational scientist at the Human Immune Monitoring Center at Icahn School of Medicine at Mount Sinai. And this video is going to walk you through a short tutorial on how we can use Clustergrammer2 to explore a single-cell mouse organogenesis atlas, actually on the cloud using MyBinder. So we're going to start off with the Clustergrammer2 notebooks GitHub repository. And if we scroll down, we'll see we have several examples and tutorial videos walking through different data sets. And we're going to go to the 5.2 notebook. So for this, we're actually going to start up an instance of Jupyter Lab, which we've already spun up over here on this tab. So you can see that it's running on MyBinder. And we're going to explore a data set from a project called the Mouse Organogenesis Cell Atlas. It was recently published by Cow and Spielman recently in Nature. So in this project, they assayed approximately 2 million cells from 61 different mouse embryos across different developmental stages, so like different days of development. And the data can be obtained via the download page. And here's a link to the paper. So for our purposes, in order to use Clustergrammer to get an overview of the entire data set, we went ahead and performed some downsampling, which I'll explain as we're running this notebook. So if we go back to our MyBinder instance, we can see our different notebooks, open up 5.2, and go to Run, and then Run All Cells. So while it's running, I'll explain how we process the data. So we broke, we first removed suspected doublets, which was documented in the paper. So that reduces the number of singlet cells to approximately 1.3 million. And here we're looking at a distribution of the 38 different cell types that were identified. So we can see which cell types are particularly rare in the data set and which are much more common. So for our purposes, to get a downsample view of the data, we went ahead and averaged the expression of each cell type in each of the 61 embryos, which resulted in a data set of approximately 2,000 clusters. Yeah, so about 2,200 clusters representing the 1.3 million cells. And we've reduced the dimensionality of the data such that we're only visualizing the top 250 variable genes across our data set. So this gives us this matrix that has 250 rows by 2,200 columns. And each column represents the average expression of a particular cell type in a particular embryo. So if we hover over the column, it'll tell us the cell type and the embryo number and the developmental stage. And we've added categories for each of the columns for cell type, developmental stage, the actual embryo ID number, and the number of cells in each of the populations. So if we look at this last value-based category, we can actually see which cell types have very large numbers of cells and which are particularly rare cell types. So the advantage of this visualization is really twofold. One is that it allows us to summarize a very large data set that is over 1 million cells in something that's a little bit easier to handle from a data perspective. But secondly, it also acts as a sort of density-dependent downsampling technique that allows us to get a visualization that's not overwhelmed by very large population cells, but that sort of more evenly shows the variation across our data set, such that we can visualize very rare cell types side by side with very not rare cell types, very common cell types. So with this visualization, we're performing unbiased clustering, and we can immediately see that the different clusters of cells cluster by cell type. So we see, for instance, here that all the endothelial cells are clustering next to each other, and we can see which genes are differentially expressed in them. So we can zoom in, see specific genes, and if we don't know what the gene is, we can just hover over the gene name, and we'll get the using the Harmonizome API, we'll immediately look up the full name, a description, and the ref-seq. So this can help us interpret our data. And we see that there's an enormous amount of structure in our data set here. So over here on the right, if we hover over the dendrogram, we can see that we have several different types of neurons, like sensory neurons, postmitotic neurons. They cluster near each other, and they have shared gene expression, which is to be expected. In this section, we have myocytes and cardiac muscles, which is to be expected too. We see, if we zoom into the cluster of differentially expressed genes there, we see genes that we would expect, like titin, which is a very abundant protein in striated muscle. And this basically allows us to get a nice overview of what's happening in our data set. And we can try different transformations. So we could rank all of our clusters by cell population size and see particular patterns in our data set, or we could choose a gene of interest and let's say NTM and rank all of our clusters by that, and then see distributions. So say we can now start to make the association that this particular gene, NTM, appears to be highly expressed in neurons, which is based on the name and the description is what we would expect. So this view is giving us kind of an overview of our very large data set and allowing us to find unbiased structures in both in both the clusters of the cells and also the co-regulation of the gene expression data. But we've also allowed you to pick a cell type of interest, and here we've gone with sensory neurons. And now we're going to grab all the data from all 61 embryos for that particular cell type. And if we perform clustering here, we see that the clustering appears to be driven primarily by developmental stage, which is what we would expect. And what's nice is that if you double-click developmental stage category, you're reordering by developmental stage, and we start from E9.5 to E10.5, 11, 12, and 13. And then we can see here that we have this distinct pattern of gene expression of genes that are upregulated at early stage of development and shifting over to these middle stage development genes and then latter stage development genes. And if we zoom in here, we're looking at sensory neurons, so we would expect to see genes associated with neuronal components, which is what we see. We can go ahead and take these gene lists like tubulin 1A. So microtubules are obviously used in neurons. We can see that their expression really doesn't come on until the latter stages of development. And so there's a lot that can be explored with these data sets, and we hope you enjoy the notebook. Thank you.